speaker is Celina Kleer. She's from University of Michigan. She's a pathologist, and as you all know, she's a principal investigator of a very sort of successful lab, and she'll be talking about a really interesting emerging target in breast cancer, EZH2. Thank you, Celina.
Good afternoon. I'm very excited to be here in person at the 2021 San Antonio Breast Cancer Symposium, talking about EZH2 functions and how to target them in breast cancer. I'd like to start with a clinical case. This is a 50-year-old woman who palpated a mass on the right breast, and this is a histological image of her breast cancer. It measured approximately 1 centimeter. You can see it's an invasive carcinoma infiltrating the fat, forming nests and glands of cancer cells. At higher magnification, this small tumor is of low histological grade. The cells are forming glands. The nuclei are fairly bland, and there's very few, if any, mitotic figures. This tumor was also ER/PR positive and HER2/neu negative, lymph node negative. So based on these features, we would predict that this patient would do well with standard available therapies. However, 8 years later, she presented with a metastasis to the iliac bone. So this case shows that we were unable to detect the poor prognosis at the time of diagnosis and guide effective treatment. This case also highlights the need to understand breast cancer biology better. So back in 2003, our laboratory established the link between EZH2, a transcriptional repressor, and breast cancer. Using a cohort of 194 consecutive invasive carcinomas, we found that EZH2 is a prognostic biomarker in breast cancer. This is an example at the univariable level, how patients with tumors that have high EZH2 expression have a worse disease-free survival and overall survival. We also found that EZH2 is an independent biomarker of outcome, providing information above and beyond other known prognosticators, such as lymph nodes and tumor size. So EZH2 is a polycomb group protein. It's the human homologue of a Drosophila protein called enhancer of zest. And this is a very important developmental protein that is required for stable expression of homeotic or Hox genes through modulation of chromatin domains. EZ is involved in regulating expression of early-acting segmentation gap genes, and it functions as a transcriptional repressor. The schematic shows EZH2 as part of a multimeric complex called polycomb repressive complex 2, or PRC2, together with other members such as SUSI12 and ED during transcriptional repression. And the functional similarities between Drosophila and human polycomb group proteins led to the development of a model for polycomb-mediated gene silencing. This is, we know at this point, quite simplified, but it serves to illustrate the function of EZH2 in this process. So EZH2, again, a member of the PRC2, together with EED and SUSI12, contains the enzymatic activity of this complex, which is affected by a set domain. And what it does is it trimethylates histone H3 at lysine 27, which is a mark that is then recognized by the polycomb repressive complex 1. And through a series of reactions that involve ubiquitination and others, there is transcriptional repression of target genes. Our studies shown that in breast tissues, EZH2 expression is very low in normal epithelium, and it's also low in usual ductal hyperplasia shown here. However, EZH2 expression is elevated in cases of ductal carcinoma in situ, especially high-grade DCIS, as the case illustrated here. The arrow is pointing to the central comedotype necrosis, and is upregulated in approximately 55% of invasive carcinomas. Here's two examples of invasive carcinomas with high EZH2 and one with low EZH2 expression. Our studies showed that EZH2 overexpression in breast cancer is associated with markers of aggressive disease, including larger size, positive nodes, higher histological grade, and also ER and PR negative disease. Now, these studies have been validated over the years by multiple independent investigators. And the features that we see in human tissues are also recapitulated in cells. There we go. This Western blot shows that EZH2 protein expression is low in benign non-tumorigenic breast cells, slightly elevated in ER-positive breast cancer cells, and maximally elevated in ER-negative breast cancer cells. To directly test whether EZH2 promotes breast cancer progression, we developed a model of EZH2 overexpression to the mammary gland using an MMTV promoter. Doing this, these are carmine alum-stained whole mounts of mammary glands of 4-month-old virgin mice. And doing this, we found that EZH2 overexpression induces a hyperbranching phenotype compared to the wild-type controls. When we look at the mammary glands histologically, compared to the wild type that the mammary ducts exhibit a cuboidal epithelium quite similar to humans, EZH2 overexpressing mammary glands have excess number of cells in the lumens of these ducts, which resemble the usual ductal hyperplasia that we see in patients' tissues, with an increased proliferative activity. So by 4 months, all the mice developed usual ductal hyperplasia, some with atypia, but they didn't form carcinomas. It is possible that these mice didn't form carcinomas because we were only able to express EZH2 twofold in these mammary glands. And in breast cancer, EZH2 is overexpressed 30-fold or 40-fold or more. But what we did is we crossed EZH2 mice with MMTV new mice, which are a very well-characterized model of mammary tumorigenesis, and found that EZH2 overexpression is sufficient to significantly accelerate tumor initiation in this model. So over the years, there's been numerous very elegant studies demonstrating that EZH2 plays a role in breast cancer initiation through regulation of mammary stem cells, and also breast cancer progression through regulation mainly of processes like migration, invasion, as well as other protumorigenic functions, including immune regulation and cancer cell differentiation. And subsequent to our studies showing that EZH2 is overexpressed in breast cancer and also prostate cancer, EZH2 was found to be overexpressed or mutated in multiple human malignancies. The prevailing view has been that EZH2 exerts its oncogenic functions by transcriptionally silencing tumor suppressors, such as the one listed here. So this prompted the development of EZH2 inhibitors with the goal of modulating EZH2 methyltransferase activity. So there's been over 35 clinical trials since 2016 using EZH2 inhibitors. And in 2020, the FDA approved an EZH2 inhibitor called tazemetostat for the treatment of metastatic or locally advanced epithelioid sarcoma and also for refractory follicular lymphoma. EZH2 inhibitors largely bind to the catalytic set domain of EZH2, but there's also compounds that bind EED and disrupt the binding between EZH2 and EED, thus affecting EZH2 methyltransferase activity. There's also compounds that induce degradation of PRC2. This is a comprehensive table that shows that there are 24 ongoing clinical trials that use EZH2 or EED inhibitors. And these are mainly used for follicular lymphoma, also diffuse large cell lymphoma, and epithelial sarcoma and other advanced sarcomas. There's a couple of clinical trials for patients with metastatic prostate cancer, mesothelioma, and urothelial carcinoma. I highlighted the 5 clinical trials that also include advanced solid tumors, with the hopes that there's some breast cancer patients in those trials. Most of these trials use the FDA-approved tazemetostat EZH2 inhibitor, which is given orally, but there's also other inhibitors, such as valmetostat, which inhibits EZH1 and EZH2, and also MK683, which inhibits EED. And you can also see that most of these EZH2 and EED inhibitors are used as monotherapies, but they can also be used in addition to other therapies, for example, with steroids, with chemotherapy, with doxorubicin, and also with immune checkpoint inhibitors. So far, there are no conclusive data from these clinical trials for breast cancer patients. I think in large part is because there are very few breast cancer patients enrolled, and there's no dedicated clinical trial for EZH2 inhibitors. Now, in animal models, EZH2 inhibitors were shown to reduce primary tumor growth and/or metastasis in triple-negative breast cancer models, and also in luminal B breast cancer models by several investigators. They have also been shown to increase the efficacy of chemotherapy in TNBC. There's been an interesting study shown earlier in the meeting demonstrating that EZH2 inhibitors enhance the efficacy of paclitaxel in triple-negative breast cancer models. They have also been shown to modify tumor differentiation of triple-negative breast cancer towards a more luminal phenotype in very elegant work performed at the Min Jae Hung laboratory, and this increases the sensitivity of these tumors to tamoxifen. And EZH2 inhibitors were found to enhance the efficacy of anti-HER2 monoclonal antibodies as well. In general, in these studies, the mechanism of action of EZH2 has been H3K27Me3 transcriptional repression. However, there's been a couple of studies that caught our attention in the lab. One that showed that although the EZH2 levels in triple-negative breast cancer are high, the global levels of the trimethylation mark are low. And another study showing that irrespective of the molecular subtype, the combination of high EZH2 expression and low trimethylation mark portends worse overall survival for patients. So this led us to hypothesize that in triple-negative breast cancer, EZH2 may be upregulated and may be functioning through H3K27Me3 independent mechanisms. We reasoned that understanding these mechanisms is important because they may influence the effectiveness of EZH2 inhibitors in breast cancer and also may offer other therapeutic targets. We found in our studies in tissue samples that EZH2 colocalizes or is co-expressed with P38 MAP kinase in a large percentage of breast cancer metastases. And P38 MAP kinase is an oncoprotein in breast cancer. This is a really interesting signaling pathway that connects the stimuli from the extracellular microenvironment into a biological response. This pathway gets activated by growth factors, stress, cytokines, hypoxia, which lead to activation of upstream kinases MKK3 and 6 that phosphorylate P38. And phosphorylated P38 phosphorylate other targets that exert cytoplasmic functions, especially migration and invasion, but also goes to the nucleus and upregulates transcription. There are 4 isoforms of P38, but in the breast and in breast cancer, the most abundant is P38 alpha. So guided by our tissue studies, that the proteins are co-expressed in metastases, we investigated whether EZH2 and P38 alpha bind. This image is from a quantitative biolayer interferometry study using recombinant proteins, showing that indeed they bind and that the binding is strong. So this led us to our working hypothesis. EZH2 and P38 bind, and we hypothesize that EZH2, being a methyltransferase, methylates, perhaps in collaboration with other PRC2 members, EED or SUSI12, methylates P38 and may activate this protein, and that P38, being a kinase, phosphorylates EZH2, and that this reciprocal interaction may operate in a subset of aggressive triple-negative breast cancers. So preliminary data from our lab that was shown earlier at the meeting in a poster discussion session shows that indeed EZH2 methylates P38. Using LC-MS, we identified 2 specific methylation sites in lysine 139 and 165, and confirmed this using methyltransferase activity assays in vitro and also in triple-negative breast cancer lines, and we're actively investigating that mechanism. Interestingly, P38 has been shown to phosphorylate EZH2 in muscle progenitor cells, but the presence of this phosphorylation and the biological relevance to breast cancer was unknown. So we set out to study this, and the first thing we did was to develop an anti-phospho-EZH2 T367 antibody to detect this phosphorylation of EZH2. And using this antibody by Western blot, we found that phospho-EZH2 is expressed in ER-negative breast cancer cells compared to ER-positive and non-tumorigenic breast cells. We set out to stain our tissues. Being a pathologist, always very excited to stain our tissues with our new antibody, and made some interesting observations. When we look at the normal breast epithelial cells over here and here, phospho-EZH2 is largely negative, but when it's present in the nuclei of the cells. However, in a subset of invasive carcinomas and metastases, we found that phospho-EZH2 is localized to the cytoplasm of these cells. When we analyzed all our data in tissue samples, we found that high cytoplasmic EZH2 is associated with higher tumor grade and with triple-negative breast cancer status. We also found that phospho-EZH2 is inversely associated with a histone trimethylation mark in invasive carcinomas. So I'm showing you here 2 examples of invasive carcinomas. This one has low phospho-EZH2 expression and high H3K27Me3, and this invasive carcinoma has high cytoplasmic phospho-EZH2 and very low, perhaps a couple of cells positive for H3K27Me3. So to investigate this from a more mechanistic manner, what we did is we used a doxycycline-inducible system based on a mutation of this upstream kinase that I showed you before called MKK6, and the mutant is EE. So with addition of doxycycline, we observed activation of P38 as expected, and we also observed increased levels of phospho-EZH2. With this, we also found that EZH2 is localized to the cytoplasm of breast cancer cells. So activation of P38 increases EZH2 accumulation in the cytoplasm. Now, if we mutate the phosphorylation site by mutating the threonine to an alanine that cannot be phosphorylated, EZH2, shown in green, is exclusively nuclear, showing that this phosphorylation event is required for EZH2 cytoplasmic localization. And to investigate the biological consequences of the phosphorylation of EZH2, we developed an EZH2 knockdown rescue system. And we did this because, as I showed you, EZH2 expression is high in triple-negative breast cancer cells. So to avoid the contribution of endogenous EZH2, we first downregulated EZH2 using shRNAs, and then rescued either with wild-type EZH2 or with a phosphodeficient mutant. Using this system, we found that the phosphodeficient mutant is unable to rescue migration and rescue invasion compared to the wild-type EZH2. In vivo, we set out to develop orthotopic xenografts with this cell line model. And I'm showing you here 2 time points at day 7 and day 63. So MDA231 cells form large tumors. EZH2 knockdown reduces tumor growth, and we have shown that in previous studies as well. But what I want to draw your attention to are these 2 conditions. So wild-type EZH2 rescues tumor growth, and so does the phosphodeficient mutant. So this means that the phosphorylation of EZH2 is not required or not important for tumor growth. However, it is important for breast cancer metastasis. While the wild-type EZH2 rescued cells form numerous metastases, the phosphodeficient mutant form much fewer metastases, which are quantified over here, and also improve survival of mice. So the next question is, how does phospho-EZH2 enhance metastasis in triple-negative breast cancer? What is the mechanism? So we hypothesized that once in the cytoplasm, EZH2 may interact with regulators of cell migration and invasion, and set out to elucidate which are these interactors. So for this, we performed an unbiased proteomics analysis and found that there's 45 proteins that interact more robustly with EZH2 wild type than with the phosphodeficient mutant. And if we look at the functions of these proteins, they're actin binding, involved in cell-to-cell adhesion, cell junction, etc. We started to focus on the actin-binding proteins because of the phenotype that we obtain in vitro and in vivo. And of those, our initial studies are with vinculin. Vinculin is a very important protein in focal adhesion formation. So by immunofluorescence, we showed that phospho-EZH2 and vinculin are colocalizing, shown here in the cytoplasm of breast cancer cells. These are all triple-negative breast cancer cells. And we were intrigued by an accentuation of this colocalization at the leading edge of these cells. As I mentioned, vinculin is an important protein in focal adhesions. So we evaluated the effect of phospho-EZH2 in this process. So while wild-type EZH2 induced multiple molecules of vinculin at sites of focal adhesions, the phosphodeficient mutant induced much fewer molecules of vinculin at these sites. And another piece of interesting data is by using proximity ligation assays. These assays detect proteins that are less than 40 nanometers apart, so basically proteins that are interacting. And this, when there is interaction, it is shown as red dots. So here we can see in this assay that vinculin and phospho-EZH2 are interacting largely in the cytoplasm of breast cancer cells, but vinculin is not interacting with other PRC2 members, EED or SUSI12. So this interaction and this mechanism appears to be independent of PRC2. So this data led us to formulate a working model for EZH2 function in a group of triple-negative breast cancers. In normal breast epithelial cells, EZH2 is large in the nucleus at low levels, where it regulates gene transcription through H3K27Me3-mediated mechanisms. And in this way, EZH2 regulates cell type identity and cellular memory. In breast cancer, there's stimuli from the tumor microenvironment, including hypoxia, cytokines, growth factors, that lead to upregulation of P38, which is a stress-activated kinase. And P38 phosphorylates EZH2 and leads to EZH2 accumulation in the cytoplasm, where it is free to bind to cytoskeletal regulators of cell migration and invasion. So this data led us to more recently hypothesize that perhaps then combined inhibition of EZH2 and P38 activities may affect triple-negative breast cancer neoplastic functions. So these initial data are in vitro. We used GSK 343 to inhibit EZH2 activity and SP202-190 to inhibit P38 activity. The red shows the combination of both drugs. And in the Western blot, we're showing that combined inhibition of both drugs reduces phospho-EZH2 more significantly than each drug in isolation, and also leads to significant downregulation of a phospho-P38 target, phospho-HSP27. But more importantly, the combination significantly reduces proliferation, cell invasion, and migration more than each inhibitor in isolation. Very recently, we performed in vivo studies to look at the effect of this combination. And so there are two different experiments. In this experiment, we developed mammary orthotopic xenografts and treated them with inhibitors. In this case, we used EPC 346438, which is tazemetostat, the FDA-approved EZH2 inhibitor for patients, and found that the combination treatment reduced tumor size greater than each treatment in isolation. And in a metastasis assay, we found that combined inhibition was successful in reducing lung metastasis in this model. So at this time, we are validating these findings in immunocompetent mouse models. This slide summarizes the current knowledge on non-canonical oncogenic functions of EZH2 in breast cancer. EZH2 also was found to have non-canonical functions in other really aggressive tumors, such as glioblastoma multiforme in the brain. So one asterisk marks the mechanisms that have been described in ER-positive models, and two asterisks in TNBC. So the non-canonical functions can be classified as phosphorylation-dependent functions, non-histone protein methylation, or transcriptional activation. As far as the phosphorylation-dependent functions, in addition to the data that I've shown you from our lab, that P38 phosphorylates EZH2, sends it to the cytoplasm to interact with proteins that regulate migration and invasion. There's been really elegant studies showing that EZH2 is also phosphorylated by different kinases. Largely studies that come from MD Anderson, and that these phosphorylation processes regulate EZH2 transcriptional activity. As far as the non-histone protein methylation, EZH2 was found to methylate ROR alpha in ER-positive breast cancer, and this induces the formation of a methyl dedron, which activates ubiquitin ligases in the cells. And I've briefly shown you our new data that EZH2 also methylates P38 in triple-negative breast cancer. What we're finding is that this methylation event leads to enhanced activation of P38 and invasion. And importantly, EZH2 was also found to have transcriptional activating functions by binding to REL-A and REL-B, activation of NF-kappa B, and also activation of Notch transcription, and by binding to SWISNF and TRIM28, EZH2 was found to transcriptionally activate CXCR4 and 12. So in conclusion, EZH2 is a promising target for breast cancer therapy. We have to recognize that when it's overexpressed, as it is in some breast cancers, it functions through H3K27Me3 transcriptional repression, but also via independent mechanisms. Elucidation of these EZH2 oncogenic mechanisms in different breast cancer subtypes is necessary for effective targeting. Mechanistic-based combination of an EZH2 inhibitor with other treatments, such as I have shown you, for example, with P38, or perhaps with chemotherapy, may be an option and a future direction. And it's also important to screen for predictive biomarkers, which will allow better selection of patients for EZH2 targeted therapy. And with this, I want to thank you very much for your attention.